Good afternoon and welcome everybody to today's webinar, the NIH, SBIR, and STTR program, Proposal Preparation Essentials. This is part 2 of a 3-part NIH webinar series. The first webinar, the introductory part of the SBIR program at NIH, is available for viewing. If you want to go back, we'll show you that, how to get there a little bit later. And then we also have a part 3 coming up next week, and that'll be available for registration. So just want to put that out there. This is part 2 of a 3-part series. I'm Robert Brook. I'm the Director of Federal Funding Programs here at the Center for Innovative Technology. CIT is a state-funded nonprofit creating technology-based economic development strategies to accelerate innovation, imagination, and the next generation of technology and technology companies. We were formed in 1985, and plugging the gaps at the earliest stages of the innovation continuum is one of our main mission here, as we help entrepreneurs launch and grow high-growth technology companies and create high-paying jobs for the future. 3 main funding programs here, in addition to some other work we do, but the funding programs are the sort of the context of what we're doing today. And we have an investment seed fund, so we invest 50 to 100K in early stage high growth potential companies for those who are in the life science biotech sector. That is a target sector for us. We have a separate investment director who runs that program. So if you have interest in that, let us know. We're happy to make those connections. The Commonwealth Research Commercialization Fund is a grant-based fund, so it's a non-dilutive funding source. There are 50 to 100K grants for both public sector and private sector. For the private sector, includes a 50K SBIR matching grant or a 50K STTR matching grant. And there's also what we call the commercialization grant of 50K. And so those are available. There's an annual solicitation. Typically, the solicitation is announced in the fall. Deadline submissions is typically late January, and awards announced in June. So for those who are looking at next year, please keep tuned with that. I will announce these opportunities in my monthly newsletter that everybody here who's registered will be enrolled in. And finally, the Federal Funding Assistance Program. And by the way, these are all available only to Virginia-based companies, since CIT is funded by the Commonwealth of Virginia. Our Federal Funding Assistance Program, we work with SBIR applicants across the state. We have some mentoring programs, consultation mentoring, low-cost proposal training and review courses. Like today's webinar, we've got full-day workshops across the Commonwealth. We have a small I work heavily with our investment team for those companies that are needing to have some sort of discussion about other non-federal funding, more of the venture capital side, so we can have those early talks. The sooner you do that, the more aware you are of the process and timing and such. We have a small grant fund that provides small awards to companies, if approved, to help you hire an outside consultant, such as today's consultant who's going to be teaching the session today. And there is an advanced application required. If you have interest in that, let me know. I'm happy to send that information to you and discuss that on the phone and see if it's a fit. And my contact information is here as well. But as far as upcoming training, we do have a couple of live workshop training coming up in April. For those who are listening to this live, it's April 2019. And the 24th and 25th of April in Falls Church, Virginia, and Charlottesville, Virginia. We also have a DoD focus sessions planned in May in Norfolk, Virginia, on the 14th of May, as well as May 15th in Fairfax. We have as I mentioned to you earlier, part 3 of the 3-part webinar series focusing on the budget next Thursday, March 7th, 2019. So these events and other events are located on our website, cit.org/events. And we have a lot of pre-recorded webinars. We will be recording these sessions here and uploading them. We have a series on NSF, DoD. We held sessions on NASA and energy recently, and we have some talks from our investment directors. So there's a lot of, so look at the URL down below, and we will be getting these slides to everybody. So if you don't have that URL at the bottom in time, you can't take a picture of it with your camera or whatever, we will send this out so you have that to refer to later. And we will also be sending out today's instructors presentation in a PDF format after the event today. So with that, I'm going to go ahead and load up webinar, excuse me, the slides for Andrea. Andrea is with BBC Entrepreneurial Training and Consulting. She is a senior principal consultant with over 25 years of experience in leading international collaborative research projects. Prior to joining BBC in 2006, she was responsible for developing a portfolio of agricultural biotechnology projects at Michigan State University's Institute of International Agriculture and for reporting to federal funding agencies about the program. She has also served as an international consultant on a broad range of issues relating to international agricultural development, biotechnology, biosafety, intellectual property rights, and technology transfer. She holds a PhD in plant pathology from Reading University in the UK, a master's in plant pathology from Cornell University, and a master's of arts and natural sciences from Cambridge University in the UK. BBC is one of the leading SBIR consulting companies in the country. We use them pretty heavily on our webinars and training sessions and refer companies to them quite often. So with that, you know, I know Andrea will talk a little bit about what they do, but we have a packed day today. For those who may have any questions, you can use the questions feature on your GoToMeeting pack software that should be on your machine to ask those questions. I will do my best to answer the questions at the appropriate time. It's not always right when you type them. It's not an interact. I don't have any long ongoing back and forth conversations with you. We'll pose those questions. For some reason it's not appropriate or we don't get to it. We can certainly answer that offline, and we can connect everybody here so that there's a resolution to any question you may have. And in accordance with time, we'll probably go a little past the 2 o'clock time today. We apologize. We will, if you need to review this webinar again later, let me know. I can send you the link to view the webinar. If you need to leave early, let me know. We can always get you that. So with that, Andrea, take it away.
Thanks, Rob. So yes, I'm with BBC ETC. This is what we do. We have a website which you can go to and take a look at and learn more about us. And at the end of the presentation, I'll show you how you can get in touch with us. Obviously, you can send an email, but we do have a form on our website that you can fill in that will trigger a process involving somebody giving you a call to talk about your SBIR plans or any questions that you have. So the website is bbcetc.com. Okay, so those of you who listened to the last webinar, we were talking about the NIH, introducing the NIH, how to approach the NIH, kind of understanding the inside workings of the NIH, and we looked at proposal review process. Understanding that we want to know what the reviewers are looking for, how the review is carried out, to understand what we need to put in these proposals. And we ended right at the point where we were about to write the proposal. So that's what we're going to do today. We're going to look at the content of the proposal and go through and take a look at each of the sections. Now, I'm not going to look at every single section because that would take a whole afternoon or a whole day. Sometimes it takes that long. But we're going to kind of look at the major pieces and kind of show you what the other pieces are. But first, to start where we ended last week, which was looking at the review criteria. So there are 5 major review criteria for the NIH, and they are significance, investigators, innovation, approach, and environment. And in your proposal, you need to address each of these and make sure the reviewers have the information that they need to give feedback on each of these review criteria in your proposal. Because this is an SBIR, significance also includes commercial value as well as technical merit. So that's important to remember. And again, those of you who did listen to the webinar last week where we talked about these review criteria in a little more detail, you'll see how they come into play when you actually look at the proposal that you have to develop here. So these are the components of an NIH SBIR or STTR. As I talk today, when I use the word SBIR, I almost always mean SBIR and or STTR, unless I say otherwise. So these programs are almost identical, apart from a couple of programmatic administrative kind of issues. So everything else I say will apply to both. This is what you need to do. So I don't know how many pieces this is here. A phase 1 proposal, generally when it's compiled and put together, it's generally around 65 pages long. A phase 2 proposal tends to be around the 100-page mark because there's more that goes into the phase 2. So that's kind of roughly how long this proposal is. You can see in the top left here the specific aims and research strategy sections. Those are the technical sections of the proposal. Everything else is administrative, background information, but it's all very, very important in terms of addressing those review criteria, because the review criteria don't just look at your research plan. So the research plan is kind of the significance innovation approach, right? It doesn't include the investigators or the environment. So occasionally people will say to me, hey, can I just send you my research strategy for review? And can you just comment on that? Well, it's generally kind of not what we do as consultants who are trying to review SBIRs, because that's not what the actual reviewers will do. They will look at the entire proposal, because if I start reviewing your research section, I'm going to have questions about your budget. I'm going to have questions about your team. I'm going to have questions about where you're doing the work, etc. So every single part of this proposal is important, and you need to give yourself enough time to get all this stuff done. This is why it takes a long time. I have a little timeline at the end of today just to scare you a little bit. But okay, let's begin at the beginning with the introduction to the application. Now, this is kind of an odd section here, but I will mention it just in case you think it's something else. So this is not for somebody who's submitting for the first time. It's not actually an introduction to the application at all in that sense. This is an introduction to a resubmission of a proposal. So just briefly, there's a lot more details here that we don't have time to go into, but you are allowed to resubmit a proposal. So you can submit a proposal. If it's not funded the first time, you can resubmit. But when you resubmit, you have to address the reviewer critiques. So the way that you do that, obviously, is to change the proposal and improve the proposal itself. But you're also asked to provide a one-page introduction to the resubmission, which is called the introduction. And in this one-page document, which is extremely difficult to write, you are asked to address the criticisms of the reviewers and to explain to them how you've changed and responded to these criticisms in the revised proposal. Again, this is one page. It can be very difficult page to write. So we do review a lot of these when people are resubmitting. But anyway, that's the introduction to the application. It's only for resubmissions, not for just a regular first-time application. So the next part is the specific aims. You'll see that's one page, whether it's a phase 1, a phase 2, a fast track, a direct phase 2, whatever you're doing. The specific aims is always only one page. That's your page limit. Now, I'm going to spend, I don't know, quite a bit of my time today talking about this one-page document. And you might wonder why. It's because we consider it to be very, very important, and the place that you should start when you're writing your proposal. I had a discussion yesterday with somebody who tells me that they write this last. We work the opposite. We definitely advise people that they start with the specific aims, with this one page. Why? And I've got a few slides to kind of explain this. It's essentially an executive summary of the proposal. We believe this is always the place you start. You don't leap into all the other parts until you've got this right. This is by far the most important part of the application. It provides an overview of your entire project from beginning to end. It tells your whole story, or it should do if it's written well. It should be compelling. So by the time reviewers get to the end of this page, it should be very, they should want to fund your project. They should understand fully why they should fund your project and how important it is. It does have to capture their imagination. It does have to tell that story that is going to draw the reviewers in and go, yep, I get it, I understand, and I understand why this work is worthwhile and how big a problem it's going to solve and why the NIH should fund this. So this one page should be able to convey all of that information. Again, it's a tough page to write. But we do view it very much as a template or a master plan for the rest of the research sections of the proposal. It should include everything about your application that is important and exciting without the detail. So it's an executive summary. It is a summary, so you're not expected to go into a huge amount of detail, but you do have to give enough detail. So it's no good just saying, well, that part is in the research strategy. I discussed that in the research strategy. If it's important to the proposal, if it's a big part of your story and why you're doing what you're doing, it needs to be in this one page. This is a page that is used by the scientific review officer to recruit reviewers for your application. So if you remember, if you were listening last week, the review process at the NIH, you submit your proposal to the NIH and it goes to a scientific review group, and the person who manages the scientific review group recruits reviewers for your application based on the pool of reviewers that they have. So he or she will look at this, decide who is the best reviewer for this application. That's important. But maybe one of the most important things to understand about this one page is that because of the way the review process is structured, your proposal will be reviewed in detail by a minimum of 3 individuals, probably by 3 individuals. Those individuals are tasked with reviewing your entire proposal in detail and giving scores for all of those review criteria and giving you an overall impact score. But then if your proposal gets through that first triage of review, it goes to the study group as a whole, and the 25 or 30 people in that room who are in that group review process, they will not be reading your entire proposal. They don't have time to, because they have a pile of proposals of their own that they were primary reviewer on. So what happens in that group is that your primary reviewer or reviewers will present your proposal to the group. The other people in the group will have access to your proposal, but all they will really pay attention to, because they don't have a lot of time, is the specific aims page. So this is the page. This is why you need to get as much as you can across and clearly and in a compelling way in this one page, because every single person in that room, whether they have given your proposal a full detailed review or not, gives you a score, and their score is worth as much as your primary reviewer's score in terms of what the ultimate review score is. So another reason why this page is so important. One thing that I do like to say here, and when you listen to my bio, you'll have noticed that I don't have a background in health. My background is in agricultural biotechnology. So why am I doing this, and how can I do this? Well, one of the, I have been with BBC now for 11 years, so I've picked up a bit about NIH on the way. But essentially, one of the things we do try and coach people on is that never assume that your reviewer is an expert in what you do. Now, obviously, reviewers are picked because they are experts in a field, in an area, and because they are good people they have some skills and background in the area. However, most of the projects that we see are very highly specialized. So you simply cannot and should not assume that the reviewer who is reviewing your proposal is an expert in exactly what you do. They are asked to evaluate grants that are not precisely in the research area. That happens very, very often. And so we and the NIH recommend very strongly that your proposal is written so that any competent scientist in your general scientific area can evaluate the application. We actually talk about targeting educated lay people. So this is not explaining this to somebody who works in your lab or in the lab next door. This is explaining it to somebody in another field. That's how this page should be written. That's how, in fact, the entire proposal should be written. So me, with my agricultural biotechnology strange background, should be able to understand your NIH proposal. And if I can't, I will keep asking questions until I can, to make sure that that information comes across. So the educated non-expert, we do recommend you avoid jargon and acronyms. I'll mention that a little bit later. And tell your story. The most important thing about this page and the proposal as a whole is that your story comes across. And there are many ways to hide that story with a lot of jargon, acronym, and too much technical detail in the wrong places. So when you Google for NIH research scientists, this is the kind of picture you come up with. So think of these people as your reviewers. You have to convince these cranky, miserable scientists. Okay, so we have an outline in BBC, we have outlines for every single section of the SBIR proposal, budgets, administrative sections, specific aims, everything. So, but this is our outline for the specific aims page. This is our secret sauce, I guess. Well, it's not secret now because it's on a slide and you can see it. But this is the outline that we bully our clients into following. So this outline is taken from the review criteria. It's based on instructions that the NIH gives. It's based on instructions that the NIH gives to reviewers and how they should review a proposal. It's based on the solicitation. So this is not from nowhere. That we've developed this over the years and refined it to make sure that this one page includes everything the reviewers need to see, and not only that it's laid out and presented in a way that makes it very easy for the reviewers to see. And we talked in the last webinar about how important it is that the reviewers like your proposal and can look at it, and it's not unpleasant to look at, that it's readable. And again, I'll mention that just a little bit at the end of today. We're going to go through each of these things in turn. Again, this may seem like a long time to spend on a one-page document, but this process is very important in the way that we work with our clients. And for us and the way we work with clients, these documents go back and forth several times. This is not a one-off review. Typically, this is often a review, a discussion, and so on until we're all happy with what comes out. One of the things we stress in proposal development, or any proposal you're developing, but particularly in this one-page specific aims, is to use headings. And we give you, as you can see in here, the subheadings for headings and subheadings for each of these sections, and we actually do mean that you should use them. We're great believers in headings and subheadings as ways of organizing the proposal to help you to make sure that you're putting in all of the information that you need and that the reviewers need to see, but also it helps the reviewers to find the information. And again, busy, stressed reviewers who have a lot to do, a lot of proposals to review, a simple thing like subheadings and headings can really make things a lot easier for them to find. So we do recommend that you use all of our subheadings as bullets, indent, bold, whatever you need to do to make this obvious. One other thing, we do recommend that you don't go crazy with numbering. People who've written a lot of other NIH proposals often use a lot of numbering. It's not really necessary at all, but certainly don't use it to the, if it's 1.1, fine, 1.A, fine, but if it gets to 1.1.2.A, then stop, because it just gets confusing. So briefly, each of these sections in turn, just to look at what should be in this page, what is the story that you should cover. So we recommend that you have an introduction. It's a short paragraph. And in the first part of this, we recommend that you introduce your company in one sentence. This is not an essay. It goes into a lot of detail. This information is in other places in your proposal, but we find it very helpful to kind of introduce yourself right at the beginning here, so the reviewers know who you are. So a simple starting sentence here would be: Small Farm Tech is a startup medical device company formed to commercialize technology developed in the Med Center at Midwest U. Simple, one sentence, just set some context here for who you are and where this technology came from. The next sentence here should tell us what you're doing. So people find this a very difficult sentence to write. It seems obvious, it seems straightforward, but people find this very difficult to do. What are you doing in this SBIR? Tell me in the first sentence, in the second sentence, really. And this should be a sentence, literally another sentence. So we want to know what your goal is. And my outline starts with the very simple words: the goal of this SBIR is. And we see all kinds of things written after that is, if people even write that the goal of the SBIR is, but people want to study, investigate, determine all kinds of things. None of those are correct in an SBIR, because the goal of an SBIR is always to develop a product that solves a problem. So that goal statement should tell us both what your product is and the problem it is going to solve. So yes, again, it's never to study, to investigate, to evaluate anything. It is always to develop a product. So write out at the beginning there, SBIR is to develop products. So tell us in your first statement what the product is and what problem it is going to solve. So you've set the stage here. We know where we're going. So now you're going to give us some background give us some context for this. Tell us about how important this issue is. And that's significance. And those of you paying attention will realize that significance is a review criteria. So this is where you kind of give the summary of how important and how significant your project and technology is. And this is where you do it. So significance is hugely important. If you don't have significance, you don't have a fundable SBIR. So what does it mean? It's here you need to tell us what the problem is that you are solving. So we often tell people to take a step back here to give more context. Often people will have, say, for example, if somebody has some kind of drug, if they're in drug development, they've got some lead compound, and they'll start telling me about the lead compound. That is not the problem. The problem is the disease or the condition or whatever, the clinical indication that this is addressing. The problem is not the drug. The problem is not how to make this drug better. The problem is pancreatic cancer or whatever the clinical indication is. So we always want to start here with something that tells us what the problem is that you are addressing. And because this is a National Institutes of Health, that's almost always a health problem. So take a step back. Introduce your technology, give some context, tell us what it is. Because this is about significance, also, we need some numbers here. We need to understand how big the problem is, how broad it is, what is the size of this problem. So numbers here are good. Any numbers that you can pull in here with a citation or two is great. We need to understand how many people have this, how many people suffer from this problem. How many people die from this problem? Just numbers. Numbers are good here to give us a scale and a concept of how important the problem is. And really in this section, we need to answer the biggest question of all, which is why do we care? Or the so what? Why do we care about this? Why is it important? So you're setting the stage for that. You're explaining and expanding on how important this problem is, the consequences of this problem, the downside. What are the negative consequences of the problem that you're addressing? And leading on from that, you want to lead into talking about your technology. But before you do that, we need to know what's done now. So again, don't assume that people know this, because they may not. Tell us what's done now. So if there is a problem, tell us what the current standard of care is for that condition, or tell us how people are trying to solve this now. And along with that, what is the gap in knowledge here? Why isn't the current solution working? Why aren't these other approaches good enough? So you're introducing why you're doing what you're doing by telling us why what exists now is not good enough and is not working. And this is a good place to tell us what you've done already. So SBIR usually you're not coming in from scratch here. You've done something. You have an idea. You have some data. You have some preliminary data that leads you to believe that your approach is going to work. So this is where you tell us very briefly what you've done already. So introduce your technology here. Tell us why you came up with this, why this idea, why this compound, why this approach. And then introduce us to tell us why you think this is going to be better, why you think this has a chance of working where the other solutions have failed. And then very briefly, what you've done already. What data do you have? What is it that you found or saw or showed that makes you feel that your approach is going to work? And we really need to know kind of where you are now and what is the current status of your technology. So remember, the phase 1 is all about proving feasibility. So if you're writing a phase 1, you need to tell us where you are now, and then you very clearly need to tell us what you still need to do to prove feasibility. That's what's going to be in this page, that story of, okay, we've shown this in vitro, but now we really need to see if this is going to work in an animal model, or whatever it is that your feasibility, your idea of feasibility is, but you do have to be very clear and state that. If this is a phase 2 proposal, you've already proved feasibility in the phase 1, and this is where you tell us how you did that. So it's not preliminary studies anymore. It's more, okay, in phase 1, we achieved our aims in our phase 1, we were successful, and we proved feasibility in phase 1 by doing X, Y, Z, and you're going to tell us how you did that. So again, this is all still part of significance, telling us what you've already achieved. And then I'm going to look at together here the product and the long-term goal. So the product. SBIR, as I said already, is about developing a product. So here's where you tell us what it is. And it is remarkably possible to write an entire SBIR proposal without really ever telling us what the product is. That's a mistake, clearly. The reviewers need to know what the product is. My recommendation for this section is to really literally start it with the product of this SBIR will be, and tell us exactly what the product is. But that's not really enough. You know, the product from this project will be a wearable fitness monitor. So what does it do? Who's going to use it? What does it achieve? Why are you doing it? What is the point of this, and who's going to use it? Why, when, how? So you would expand that by a wearable fitness monitor to be worn by individuals with diabetes to whatever. Explain it. Who's going to use this? What is the clinical use of this? And what is it supposed to achieve? So it's going to measure what, why, how, and what is the outcome of that going to be? So tell us more than just what this physical entity thing is, but tell us what it's going to do, who's going to use it, when, why, how, and what the outcome is intended to be for the product. And again, that section it's good to start that section with the product of this SBIR is, because that will stop you writing a paragraph of stuff that is not defining the product, which people love to do. It's good here also to tell us what's novel about this. And people struggle with this section because they want here to just describe it, and that's not what this section is about. So SBIR has an I in it. That I is innovation. So you have to tell the reviewers what's novel here. What is the innovation? Why is it novel? How is it novel? How does it change the status quo? So that's why technological innovation is here. Very, very important to put that in, because again, innovation is a review criteria. So make sure that you're explaining to the reviewers how this is innovative. And then impact. So impact actually overlaps with the long-term goal. So sometimes I tell people they can just write the long-term goal. It depends. Sometimes the impact is a little bit different than the long-term goal. But essentially, long-term goal is a super important section of this one page, because it tells the big picture. And it's about impact. So the long-term goal is just that. It's long-term, it's big picture. You're asked to think ahead here. This is not about you. This is about the impact of your proposal, of your technology, of your project, of your product. So you're thinking ahead. So not 2 years, you're thinking 5, 10, whatever, 20 years ahead here. What is the impact of this is going to be? What is it going to change? Who is it going to affect? How is it going to benefit people? How much? Who is it going to benefit? So I say it's not all about you. It's not about your company or your goals for your company. It's not even about your product and what you want to do with your product. It's about the impact of your product on the problem that you say you are trying to solve. The other thing to remember here is it's not all about money. Yes, we love money. Saving money is great, but the NIH is not about saving money. That's not the primary goal of the NIH. So although you can mention money, it should never come first. It should never be the focus. You should always, where possible, be addressing your long-term goal here to improvements in clinical outcomes if you possibly can. Now, not every project lends itself easily to this, but this is the NIH. So really, to some level, you do need to make sure that the reviewers understand how your project and how your product is going to address issues of national health. So think big, make it compelling. Make it compelling because this is essentially the reason. This is a sentence that tells the NIH why they should fund what it is you're doing. And I put here, remember the mission of the NIH. So anyone who's written a grant for anything ever, whether it's to the NIH for an R01 or to a foundation for playground equipment for your school or whatever, you have to address the mission of the agency, because every agency that gives money, every foundation that gives money has a report to write at the end of the year, has to report to somebody who is providing them money on how they have achieved their mission, how their use of money has addressed their goals. And the mission of the NIH is to seek fundamental knowledge about the nature and behavior of living systems and the application of that knowledge to enhance health, lengthen life, and reduce illness and disability. So even if you have an upstream technology, a widget, a platform that is helping you do something that is very upstream, make better drugs, screen drugs in a more efficiently, whatever it is, all the way down to an actual therapeutic that is addressing a disease indication, your proposals must still focus on how it is going to enhance health, lengthen life, reduce illness and disability. So make sure that in everything you're focusing on that mission, and that should be your long-term goal. Then we finally get to the actual kind of research part. You're going to hear that this is where you start to tell the reviewers what you're going to do in this project that you're presenting here in this proposal. So let's talk about a hypothesis and specific aims. Well, the NIH, as you may know, is essentially a hypothesis-driven agency. Almost all of the proposals that they fund are hypothesis-driven. I'm not going to explain what a hypothesis is, but essentially you have to have that research question, that research hypothesis. You've seen something, now you're going to investigate whether that thing is true. However, I'm going to just say here to introduce this that for SBIR, sometimes a hypothesis is not essential. Now, we like hypotheses. We do encourage people to write good hypotheses because we think they can be helpful. But for SBIR, if you have a clear product development plan, you don't necessarily need a hypothesis. Having said that, if you have one, you should state it clearly, specifically. It should be one simple sentence. It must be testable, as any hypothesis must be testable. We like hypotheses because generally NIH reviewers like hypotheses. They expect hypotheses, so it does help you to get things across to the reviewers. It does also, if it's written well, it can help you focus and determine what your specific aims should be. And it helps you write the proposal, because once you write a good hypothesis, it should give you a lot of information on what aims you need to have and what experiments you need to set to address those aims. So I've said the word aim several times now without really explaining what I mean. And this is something that I've never seen, that everybody struggles with. In fact, I've never seen anybody not struggle with this. There's a lot of negatives there. Goals, objectives, and activities. Okay, objectives and aims are kind of the same thing here. A goal is a longer term thing. It's where you're going. It's your general direction. It's what you want to achieve in the future. Objectives and aims are the strategy. This is the way you're going to get there. This is your plan, the way that you're going to approach reaching your goal. And activities and tasks are the things you do to achieve your aims. Now, most people know what their goal is, and then when you ask them to write objectives, they actually write activities or tasks. I cannot explain this it's hard to give examples here, but when you sit down and try and write these, you will probably struggle a little bit here. Hopefully I'll give you a little some help here to try and explain it a bit more, but it's harder than you think to write these aims. But essentially these aims, you need to have them. An NIH proposal almost always has research aims. The aims generally obviously are related. It's ideal not to have them dependent upon each other, where if one fails, you can't do anything else. They should be about 2 to 4 sentences. I'll show you a good way to structure them in a little while. But essentially they're explaining and describing your experimental approach. A typical NIH grant has between 2 and 4 aims. Typically, a phase 1 is 1, 2, sometimes 3 aims. By the time you get into a phase 2, it can be more aims than that. If there are 6 aims, you've written your work plan and your activities, not aims. Aims should have a great title, and every aim needs a milestone. Milestones. The biggest critique of NIH proposals, the strongest critique ever, the most fundamental critique that reviewers give to proposals is no quantitative milestones. You have to have, for every specific aim has to have a milestone, and it should be a quantitative milestone. These are not always easy to come up with, but they are essential. There has to be some kind of measure of success, a quantifiable measure of success in your aim. And if you have a well-written aim, it's a lot easier to see what that should be. If it's a poorly written aim, you'll find it very hard to write a success milestone for it. If you've written a task, it will be impossible to set a milestone for it. So you do need to think this through, and this is what takes almost the longest, I think, about writing this page or writing your proposal, determining what your aims are and how you're going to measure success. So quantifiable, that means there should be a number in there, obviously. And the endpoints or the milestones that you set should be meaningful in terms of the biology, the science here. It should make sense, and they should be viewed by reviewers as sensible, appropriate endpoints for what it is that you're doing. So you can't really just make them up. Now, sometimes people do, because they don't really know what their endpoint should be. But this is a very important thing to do, obviously, in any science or in anything, is to determine the point at which you need to accept that doing what you're doing is not going to work and do something else. I mean, that's essentially what a milestone is. This measure, if I get this, I've achieved it. If I get this, I haven't. And there needs to be some kind of threshold or cutoff or parameters within which there is success and outside which there is failure. And you need to describe that clearly. So this is a milestone example, the completion of a 28-day toxicology study in 2 species. I see milestones like this all the time. It is not a milestone. Telling me you have done an experiment is not a milestone. That is a bad milestone. 28-day toxicology study in 2 species with no observed adverse events at drug levels at least 5-fold above the therapeutic dose. That is a milestone. It's not perfect, but it's better. It has some numbers in there. If you get less than that, less than 5-fold, then you have failed. If you get above 5-fold, you have succeeded. There is a measure, there is a threshold, there is a success and failure marker. Again, medicinal chemistry optimization will produce compound analogs with improved potency and reduced toxicity. So many red flags in here. Optimization, what does optimization mean? What is the end point of optimization? What does improved mean? What does reduced mean? There is no way you can say you have achieved this. There is no measure of success here at all. That is a very bad milestone. Medicinal chemistry optimization will produce a compound analog with EC50 less than 10 nanomolar and an LD50 greater than 5-fold above the EC50. That is a milestone. Again, you can see what success is. You can see what failure is. It's obvious. That's a good milestone. So a good specific aim. This is I'm sure you can write a specific aim that isn't exactly like this, but a specific aim has a title, and the title is always has it includes kind of a why, not just to what you're doing. It tells you why you're doing it. It's a brief title. No specific aim should be longer than 1 sentence. That is crazy in terms of the title. Then you're going to tell us very briefly what you're going to do, how you're going to do it, and what you're going to measure. That goes in the middle of the sandwich here, and at the end of the sandwich we have a criteria for acceptance or a milestone. Tell us what your success metrics are. What do you need to see in quantitative terms to be sure your aim is successful? You need numbers. As I just said, I just showed you, you definitely need numbers in this section. Quantitative milestone has numbers in it. So you need numbers. So here's an example. Specific aim 1, confirm the tumor inhibiting activity of compound X in mouse model. So I made this up, but anyway, you're not just saying measure the tumor inhibiting activity, or look at, or whatever, or confirm tumor size even. There's a focus here, there's a goal. Confirm that the compound X inhibits tumor growth. So you're definitely showing us in this title what it is you're looking for, not just what you're doing. And then a very, very brief experimental plan. 24 mice will be injected with 25 microliters of compound X, and tumor size will be measured at 14, 24, 28, and 42 days. Control mice will be injected with sterile water. That's what you're doing. Brief, not a lot of technical detail, especially technical things that you haven't told us about in this page. Don't start introducing them here where we don't understand them. And the criteria for acceptance and milestone here is statistically significant reduction in tumor size of greater than 75% compared with the control. It's quantitative. You are looking for something better than 75%. If it's less than 75%, that doesn't matter. It's not going to be success. So you have to decide what that success measurement is going to be and put that clearly here. So phase one, I'm talking specifically about phase one here. The outcome of phase one has to be, always has to be, because that's what phase one is for, proof of feasibility. So don't forget that. So if you do, I'm sorry, this is more than I thought here, but so if you cannot write your phase one proposal without using the word feasibility or proof of concept in it. Otherwise, it is not a phase one SBIR. Phase two is a little bit different. Phase two, so this is a phase one outline we're looking at here. A phase two outline is almost the same without the plans for phase two, essentially, and where your preliminary data is really your results from your phase one. So, but the reviewers, this isn't just, although this is a phase one proposal, the reviewers want to know where you're going after this. So that we have a section here which is plans for phase two. Okay, so what are you going to do after proof of concept? And you only have a couple of sentences to do this, but be as specific as you possibly can and tell them the next R&D steps. After I've proven feasibility, this is what I need to do next to take my project into phase two. And then a section on commercialization. What is commercialization? The ability to provide a solution to a problem in exchange for money. So the reviewers in a phase one want to see a very brief detail here. In a phase two, obviously, you're expected to know a lot more about the commercialization of your product. But even in a phase one, they want to see that you understand your market. Do you know how big it is? Very basic. Do you have any numbers here? Do you understand who is going to buy this and how many of them are there? And the other thing they want to see is, what is your strategy to get it there? Have you thought that through? Do you understand how to get a medical device to market? Do you understand what partnerships you need, what regulatory issues there are? In phase one, you don't have to know everything, just show you've thought about it. By phase two, you need your act together. Okay, that's a specific aims page. Here we are, like 3/4 of the way through the presentation. That's just specific aims. But I can't get that all on one page. This is what I hear a dozen times a day. Yes, you can. It's not easy. It's a challenge, but yes, you can absolutely do it. This is just our little example of how this page should look. This is obviously not real text in here, but this is how it should look roughly, with an example of how much should go into each section here. So we show that to people when they're panicking as to what should go into this page. When I review proposals, I will review a specific aims page. It's a page and a half, that's fine. But anything beyond that, you have to try and cut it down. I can't cut down something that's 3 pages. So you can start at a page and a half and cut down. And again, this is sometimes why it takes a long time. So research strategy is the next section here. This is the main technical section of the proposal. It has 3 sections: significance, innovation, and approach. What are they? They are review criteria. So the proposal you're writing is structured to help the reviewers determine the review based on these criteria: significance, innovation, approach. They are 3 main sections. And just very briefly, I'm going to look here at we looked at the review criteria last time. Here I'm going to just essentially show you what you are asked to do. And this is in the solicitation. This is what you are asked to do in each of these sections. In the significance section, you're asked to explain the importance of the problem or critical barrier to progress in the field. You're asked to explain how the proposed project will improve scientific knowledge, technical capability, and/or clinical practice, to describe how the concepts, methods, technologies, treatments, services, or preventative interventions that drive the field will be changed if the aims are achieved, and describe the commercial potential of the product to lead to a marketable product, process, or service. So that's what should be in the significance section. And if you can remember back to 10 minutes ago when I talked about the specific aims, that's what you wrote there. That was the summary of this. So when you get to the significance section in the research strategy, this is where you start to expand on the information, the summary that you had in the specific aims section. And I did forget to say that for a phase 1, this research strategy section is 6 pages long. For a phase 2, it is 12 pages long. So 6 pages for a phase 1, 12 pages for a phase 2. There are no page limits for these, or no directions for how much should go to significance, innovation, and approach, but we have recommendations that it's about a page to 2 pages for significance, a page for innovation, and then the rest of the proposal should be dedicated to the approach section. So we have an outline to this section, which is essentially what we just talked about in the specific aims: the problem, the product to be developed, the impact, the innovation, the commercial potential, market analysis, commercialization strategy, and so on. So this is the outline that we give you. For this section, when we give you an outline, we do expect you to add your own kind of headings in here as well that are specific to your technology, because you'll probably have some other subheadings in here. But essentially, this is just an expansion of what we've talked about for the specific aims. And again, for innovation. So in this section, you're asked to state the innovation, explain how it challenges and seeks to shift research or clinical practice paradigms, describe any novel theoretical concepts, approaches, methodologies, instrumentation, etc., and to explain any refinements, improvements, or new applications of concepts, approaches, or methodologies, instrumentation, or interventions. So tell us what's new and novel and exciting and paradigm shifting, whatever, about your technology. And for the NIH, this doesn't necessarily have to be as super innovative as you might think it does. This is sometimes innovation. There has to be innovation in this. It does have to be new, but sometimes people think it has to be completely groundbreaking, earth-shattering, and that's not necessarily the case. There does have to be innovation there. You do have to describe it. And again, this section, we give you a brief outline, but you are expected to kind of add in your own subheadings here that are specific to your technology and product. The approach section, this is the meat of the proposal, where you tell the reviewers what you are going to do in this phase, whether it's a phase 1 or a phase 2, describe the strategy, methodology, and analyses, how data will be collected, discuss any potential problems, alternative strategies, and benchmarks. This is science. Stuff goes wrong. What are you going to do? Show the reviewers that you understand things could go wrong and how you're going to approach that and how you will deal with that. This is also where you point out any procedures that are hazardous to personnel and precautions. This should include also any information on your preliminary studies in a phase 1. In a phase 2, the approach section will include your phase 1 final report. So the phase 1 final report would replace the preliminary studies that are in the phase 1. So preliminary data is a section people struggle with a lot. Because they try and put too much information in here, or they cut and paste from publications. They put in graphics and charts that are very technically detailed, that are very, very small. That's very common. People put things that are much too small for anybody to see, and suddenly things get super technical, and you've lost maybe 60, 70% of the reviewers because suddenly here there's too much technology that people may not be familiar with. So a couple of things that you should do in this section is summarize your work. You're not supposed to give a lot of detail here. Your goal is to summarize it, tell us what the aims of the work were, and what you found. So we'll see the specific aims section is going to be almost identical to this. Tell us what the rationale was for those studies. Tell us what the aims were for the studies, what the results were, and the summary in terms of how does this work apply to this SBIR. So how did we get here with your preliminary data or with your phase 1? To summarize it, I like to see a heading of summary and an actual kind of statement, a couple of sentences that explain what that showed and now how that brings you here to this phase 1 or phase 2 SBIR. So that section, depending on how much preliminary data you have, obviously this section for a phase 1, for example, would be half to 3/4 of a page, maybe a page at the outside. This should not take up the major part of your approach section. So most people try and put too much in here. So summary, briefly outline results, and also you're asked to list any publications. I think that was covered pretty much on the previous slide. The specific aims themselves. So you wrote the specific aims on the front page. Those specific aims, specific aim 1, 2, 3, or whatever. Now you're going to bring those here, and you're going to tell us more about them and how you're going to do them. So here you'll have the title of the aim, a short paragraph explaining why this aim. What is important about this aim? What are you going to find using this aim? How does it fit here? Why are you doing it? A section on experimental design and methods, very straightforward, how you're doing it, what are the methods, how you're going to analyze the data analysis, how you're going to interpret the data. Remember, we talked about potential pitfalls and alternative approaches. A section on this is very helpful because the reviewers are going to be looking for this and will comment on it. They're asked to comment on this. What could go wrong and how will you compensate if it does? And expected outcomes, what do you expect to happen? So every aim should look like that: rationale, methods, data analysis, pitfalls, outcomes. Do remember, if you have a proposal or a project that involves any kind of statistical analysis, numbers, whatever, that you do plan your data analysis well, that you do have somebody on your team, a biostatistician or somebody with the ability to analyze the data. Make sure that you've set this up right. If you're doing any kind of analysis, that you have the power level that you need or whatever. There will be somebody on the review group who is a biostatistician. We like to recommend also some kind of Gantt chart at the end of your research strategy section that tells us who, when, and where. We care less about the when than we do about the who and the where. So this is a very useful section of the proposal to tell the reviewers who's responsible for what, who's doing specific aim one. Is it you in the company? Is it a collaborator in the university? Is it a consultant? Is it another kind of partner? Tell us how that's working. Sorry, trying to move the slides on here. I've gone quiet. Okay. I am not going to talk about human subjects. If you are doing human subjects, this is a very large subject that I'm not qualified and we don't have time to talk about. But do be aware that if you have research involving human subjects, there is other stuff you have to do. Obviously, there's a lot of paperwork that you have to complete, forms you have to complete. Human subject research includes everything from collection of blood from actual human beings to conducting a survey, to testing a new educational technique. So do be aware that it's broader than you might think. We often find that people developing apps for behavioral interventions suddenly trip up here and don't realize they have human subjects when they do. The NIH has a great website, humansubjects.nih.gov, that will take you through a little flow chart asking questions that will bring you through the process to understand whether or not that you do have actual human subjects in your research. Sorry, the slides have gone a little crazy here. Okay, I'm just pausing for them to catch up with me. Maybe that, okay, so the next one. Sorry, I've kind of lost control. So
let me know if you need any help on that.
Okay, thanks. I think it's, no, it's kind of, I hit too many buttons now. So, but the NIH website does have very useful kind of flow chart thing they've done now. It used to be on a piece of paper, but now it's excellent. It's gets you to put information with, into an online form that will take you through the process to decide if you have human subjects. Also be aware that there is a type of human subjects research, which is a clinical trial. And the NIH has recently changed the instructions on what is a clinical trial. You have those 4 questions to answer. And if you have yes to all of those 4 questions, then you do have a clinical trial. This is important because, so does it involve 1 or more human subjects? Are you assigning human subjects to interventions? Are you evaluating the effect of those interventions on a health outcome? I'm sorry, the slides are going backwards and forwards now. But again, the NIH is there to help you to decide whether you have a clinical trial or not. The most important thing about whether you have a clinical trial in terms of SBIR is which solicitation you respond to, because there are specific solicitations that are for clinical trials or not for clinical trials. So do be aware of that and make sure that you submit to the right one.
And those are pretty evident in the in the SBIR section of the solicitation. It indicates it like SBIR, STTR, human subjects, SBIR, non-human subjects. It pretty much is outlined like that, right?
Exactly. Yeah, it's fairly clear. The most difficult thing is deciding whether you have a clinical trial or not.
Right.
Okay. I don't know how we can get this back on track here, but
Do you want me, what are you trying to get to, this page right here, the components page?
I guess so, yes.
Okay, let me try to take it and I'll just, there you go. Hold on, there we go. Hopefully that's, okay. Okay.
I'm still, okay.
So yeah, so I've given it back to, I mean, you should be able to, maybe it's waiting
Okay.
a second, so.
Okay, the problem, I still see the clinical trial page, so I'm not seeing what you're seeing, I guess. Maybe I have a lag on this end.
I think you might. It's on the component, it's on the main components of an SBIR, NIH SBIR slide.
All right, it's hard for me to to wing it, but okay, we'll work on it. Okay, so
Now it's moved. Now it's gone to consortium contractual arrangement. So let's just, I guess it's probably just waiting. So.
okay, so are you seeing vertebrate animals? I'm seeing vertebrate animals. If you're using vertebrate animals in your proposal, also you have obviously some administrative things to deal with, some paperwork to address that. I'm sorry, what are you saying, Rob, now? Anything
We're
relevant?
what page do you want to be on?
Okay, we should be on multiple PI plan here.
Okay, hold on. There it is. Okay, I've got it.
Okay.
It's
All right.
there.
Well,
So hopefully it's yeah.
okay.
You can't just keep hitting like that back because it will catch
I
up.
know.
And
Okay, so
but I'm not sure if mine will overtake yours or not. So
okay.
I'll
Well,
keep it on the MDI plan if it goes away. All right.
you take control, and you'll just follow what I'm talking about. Maybe that'll
Sure.
help. How's that?
Sure. No
Okay.
problem. That
So
sounds great.
you can have multiple PIs on an SBIR. That is fine. Sometimes it's appropriate to do that. Make sure that if you do, that you have a multiple PI plan. So you will have to kind of explain out and have a document explaining how this is going to work and who's in charge and who makes decisions when and how you meet and some of those issues. So that's another little piece of administrative stuff you will have to do. Moving on to consortiums and contractual arrangements, this is your broader team here. So an SBIR often, almost always, has some kind of consortium, where a consortium is essentially defined as a group of institutions of some sort. So if you have a subaward, if you have a subcontract, then you have a consortium. And there is a document called Consortium and Contractual Arrangements where you can describe that consortium. This is a great way to describe your partners and also what they're responsible for. So you don't waste time in your research strategy explaining that. It goes here instead. So you're explaining the details of the relationships, the deliverables for each partner in this document. And along with that, just briefly to mention that each of these partners, whether they're an institution or a consultant, will need to provide you with a clear letter of support. And that letter of support should just be a page, very simple, verifying whatever it is they're committing to the project and explaining what it is that they're contributing in terms of, if they're a consultant, in terms of their hours of commitment and then what is it they're actually doing, if they're a collaborator, what resources they're committing. And any of these collaborators will obviously also have to provide you with a biosketch, which I'm going to talk about in just a few minutes. So yeah, letters of support from consultants are almost the same as any other partner. Just make sure the information is in there. People do like to write a lot of other stuff in these letters. That's okay. But really all people are looking for in these letters is, what is the commitment? What are these individuals committing to this project? What are they doing? And generally, we advise against the very general blah, blah, kind of these people are great, this project is great kind of letters, but really sticking to letters that are strong letters of support and commitment. Appendices, almost always, there are no appendices allowed for an SBIR. So do not add anything into the appendix section. Everybody loves to put stuff in there. They are almost always not allowed. Sometimes you will see a targeted solicitation, a different solicitation where they ask for something in the appendix, but almost always this is not appropriate. Bibliography. This is where it gets super easy. References cited. So any reference and citation in your proposal, there is a separate section called Bibliography and References Cited where you combine all of these citations and list them all here. What do people ask about this section? Well, they should be relevant. How many? More than 10, less than 300. It depends on your project, but adding more doesn't necessarily make you look smarter. Obviously, make them relevant, make them applicable, and use recent updated references in this section. There is no page limit, but again, be reasonable here. Don't go crazy. You are allowed to use any format here to do this in your bibliography section. That's fine. Whatever format is appropriate for your field. Pick a journal, pick a format that that is appropriate, and use that format for this section. Project summary abstract. Another very easy section to get done. Essentially, I like to think of this as specific aims light. So you take your specific aims and you smush it down into 30 lines of text. The limit is 30 lines. So just make sure that everything you can possibly get into that summary is there in that. That's just a one paragraph document. There's less than 30 lines of text. It will be publicly available, so be careful not to include any proprietary information in that document. Public health relevance, another easy section to get done. This is, sometimes it's called the project narrative because of the way the forms are set up, but essentially this is a 2 to 3 sentence document that describes the relevance of this research to public health. This is where you explain to a layperson, what are you doing and why? Why is this in the NIH? Why is the NIH funding this? How does this maybe upstream project address human health and address issues of human health? So it's a useful section to write. Don't stress too much about it. This is a section that's pulled out by Congress to show that the NIH is doing its job and addressing human health, essentially. So again, 2 to 3 sentences in plain language. Key personnel, let's talk just very briefly here about biosketches. Go through the main sections just so you see what has to be done. Biosketches have to be provided by anybody who is a key personnel. So that's your employees who are key personnel, any subcontractor employees, any consultants, and any other significant contributors to the project. The reviewers are looking at your biosketch. What for? If you remember that review criteria of investigators, this is where they go to judge the investigators and whether they're qualified to do the job and a good match for the project and the role that they have in the project. So the reviewers take biosketches very, very seriously. You really need to pay attention to putting these biosketches together. So just briefly looking at the format here, again, all senior personnel have to submit an NIH biosketch. There are very strict regulations and instructions on how to do this. There is a sample biographical sketch page. There is a downloadable sample, there is a downloadable template, and the most important thing about this section, about this proposal section, is that you have to do it exactly as they say. They like to see it exactly as they recommend. So do not veer at all from the format of this section. 5 pages per person, and each of these is attached as a PDF file, as all of the other sections of the proposal are. There are 5 sections to the biosketch. The first section is essentially your name, position, education, training, very straightforward. Then a personal statement, then any positions and honors, contributions to science, and research support. Looking at those sections, each of those sections briefly. Personal details, this is the easy one where you put your name in, your position title, and your education. Not difficult to do that. Personal statement, a little more challenging here. This is a paragraph or 2 that should be specific to each individual application because it should talk about how you are qualified for the role that you have on this particular project. So this is where you're supposed to focus on selling yourself and your strengths and your abilities and your experience and your background and your expertise for doing this project. So this should be tailored for each application, not just cut and paste or the same biosketch used for every proposal. The personal statement, again, should be customized for each. If you download the example that there was a link for a moment ago, it will, it gives you an example there of the kind of approach, the kind of level to approach there. It should be personal, but not overly personal. Positions and honors, section B of the biosketch here, you list your positions in employment, any other experience and professional memberships, and any honors. And again, the most important thing about this section is that you follow this format exactly. Don't add information, don't take information away. All they want to see here is your position, where it was, and what the institution was, and where that is. So assistant professor, Department of Psychology, Washington University, St. Louis, Missouri is all they want to see here, and the dates. So that's the positions and honor, and the same with any other experience, professional memberships or honors. Keep this down, not more than one line for each position or honor. Contribution to science is the most difficult part of this. So previously, all the NIH reviewers wanted to see was a huge long list of publications. They don't do that anymore. They want to see more than that. Rather than just a list of publications, they want you to summarize the things that you have done, or the individual has done in some sections here that are lumped together in contributions to science. So say, for example, myself, I worked on potato diseases in upstate New York. I worked on banana diseases in Uganda. Okay, so those are going to be two sections. I'm going to be talking about my contribution to the potato disease and breeding program where I did my master's degree. I'm going to talk about my role in that. And there were some publications that came out of that. I was not first author. I did not lead the project, but I had a role in it. And this is the section where you get to describe what your role was in that and how important it was and how you contributed. So pick 2, 3, 4 of these that you can do, set contributions to science, and list each of those in turn with a brief paragraph. Here's an example, a title, a brief title, role of plasma membrane, monoamine transporters, whatever. Tell us what you did, where you did it, how you were involved, how important this was. And then you're allowed to list 4 outputs. These are not necessarily publications. These can be any research outputs. So this is great for people who don't have a normal kind of NIH biosketch. So if you're in education, or health education, or you're a software developer or something, here you can put your software products or your curricula or whatever. You don't have to, doesn't have to be just scientific papers. Obviously you have papers, this is where you put them. But there, so you're allowed 5 contributions to science, and each is allowed 4 outputs or publications. The research support section, again, is very simple. They give you an example here, 2 or 3 sentences, where you list any ongoing and completed research for the last 3 years from any source, federal or non-federal. And briefly indicate the goals of the project and the roles of the individual, and your role on it, the dates and the role. A very simple section where they're looking here to see what your role was in projects, obviously, but how many things you're involved in now. So if you have 7 of these, 7 ongoing research projects, the reviewers might have a reasonable question about whether you have time to do this, for example. Where do you find the biosketch templates? I lose them, and every time I find them again by Googling NIH biosketch format, and it will bring you to this page that talks about biosketch format, and you can download a blank format page, you can download the instructions, you can download a sample, all at this page that's very helpful. So all of that information is available here. Here's what you download. If you download the example, this is what you'll see, the position. I'm not going to go through this because we've talked through it, but this is what it will look like. Just make sure when you're putting yours together or you're putting your teams together, they look exactly like this, with one exception: that sample watermark that goes through the back there should not be there. So please take that out before you submit your proposal. Okay, let's skip through those. Again, download the templates, download the examples. So I just want included here, so in case you don't believe that investigators care about, the reviewers care about this, I just cut and paste some examples here from review statements that I've seen where the reviewers are really paying attention to this in the investigator review criterion. So I'm not going to read all of these, but do look and see when you get the PDF. You can see that they're commenting on the PI and their current position, where they are, whether they're competent, capable, whether they have any evidence of having done this before, whether they have experience, and whatever. And then looking at the whole team. They're paying a lot of attention to this section of the proposal, so you should too. One of my favorites here is the last one, the do reviewers care, and this one, all of the bio sketches included are outdated, and much of the included information is outdated. On the bottom here, one of the bio sketches contains a sample watermark. The lack of attention to detail in the preparation of this application does not inspire confidence in this group of individuals. Don't be that group of individuals, okay? Take some time to get this right. The biggest thing to remember about bio sketches, if you're in charge, if you're the team leader here putting this thing together, make sure you get everybody's bio sketches in early. And it's your job as the project lead or the person putting this proposal in to make sure everybody has put their proposals, their bio sketches together well. Take the time to fix them, make sure they're consistent, make sure they follow the instructions, and look professional. Okay, moving on, we're almost there. Facilities and other resources and equipment. Remember the environment criterion, their environment review criterion. This is where they find it. The facilities and other resources document. There should be more than just a cut and paste list of your facilities, but an explanation and analysis and a small narrative on how this is going to benefit the project. And you should here definitely describe all of the facilities, not just yours as a small business, but any other facilities that you're using. Any lab facilities, animal facilities, computer facilities, and so on, and definitely, and all of your partners' facilities. So if you've got animal work being done somewhere, you're going to describe that. You're going to describe also the facilities of your collaborators in addition to your own lab space and office space, or whatever. And we have a little outline for this section. Actually, we have an example now that you can download based on the Star Trek universe, which is kind of funny. But always start with the company's research facility and then move on to any subcontractors. But the other thing you can add in this section, because it's other resources, this is where you can talk about other things that you have going for your company. Not just the science, but any commercialization resources. Do you have a CEO yet? Do you have any management, company management? Do you have any other funding? Do you have anybody on board who knows about regulatory, reimbursement, whatever? So you can definitely here talk up any business, management, other resources that you have in your company to show the reviewers you're not just a bunch of scientists who can't do the business side, but you've thought about it and you're starting to put together a management board, an advisory board, or whatever. So this is a great place to put that information in your proposal. And again, also you include your equipment in that, so the reviewers can see you have access to equipment. I'm not talking about budgets today, that's a whole other subject. Budget justification, though, is another narrative page or two that goes into this documentation. A budget spreadsheet is just that, it's a spreadsheet. The budget justification is where you tell the story. So it's where you should be talking about the details. So every single line item on your budget should be explained in more detail in a short paragraph in your budget justification. This is a great document for a reviewer to go to see exactly what's happening and what the money is being spent on. So it's more of a narrative than a justification, but it's a very important part of the proposal, so make sure you leave time to do it. And we have, and again, another example and a template for this section.
And again, we will send these out to everybody. So some of those areas we've kind of gone back and forth in from the slides. We apologize. There was some latent interaction between my desktop and her desktop. So sometimes that happens on these meetings. We apologize.
So we're almost there now, just a few more slides. So commercialization plans, I'm not talking about them today because again, that's a whole webinar on its own, but, and we've seen the second slide already here, but do remember that commercialization is a part here. You are going to be judged on your commercialization strategy. Even in a phase one, they're going to be looking at what information you have here, that you understand the market, and that you have a plan to get this product to the market. And it varies a little bit from agency to agency what is expected here and what's expected from phase 1 to phase 2. So the NSF, for example, require a lot more in their phase 1, a lot more commercialization information in phase 1 than the NIH do. The NIH kind of let this slide a little bit in the phase 1. So we think about it more as a kind of in the phase 1, you're providing commercial justification. In the phase 2, more of a detailed commercialization plan. The NIH commercialization plan has, I think it's 6 sections here, and with an outline, value of the project, company information, market, customer, competition, intellectual property, finance plan, production and marketing plan. And that should be no more than 12 pages. Make sure, obviously, that you get that started early. This can take a lot of time to put together well. And if you don't have those people in your team, you're going to find some people to help you put this together and do it well. So yeah, don't forget commercialization. If you build it, they will come. It's not a great strategy because you could be waiting for a long time. Okay, a few more slides. Just so you put in your great proposal, you write it, you respond to the review criteria, you put it in. We believe you should also develop a beautiful proposal. And again, reviewers are human beings, right? So 2 important things here, you must follow the rules. So there will always be rules to follow in terms of formatting and layout and how this proposal should look and be put together. Where are those rules? They are in the solicitation. So always read the solicitation, read the small print. You will find there all of the details that you need about font and typeface and spacing and headings and so on. Also remember, it's about more than just following the rules, because you can follow the rules and still produce a document that nobody wants to read, okay? And your job here is to make the reviewers want to read your proposal and not hide it at the bottom of the pile that they have in front of them. Do remember again that reviewers are not necessarily experts, so the way that you write your proposal should be educated at that, targeted at that educated non-expert. You can absolutely and should use figures, charts, and diagrams when they work, when they're appropriate, when they help you to describe something in an easier way, in a better way. They break up the text. They help reviewers to see stuff easily. Make sure they are big enough. Make sure they are clear, and definitely make sure they're big enough, because people do tend to put them in too small because they're trying to save space. But if you're using them, make sure that they're big enough to tell the story. Be aware also that not all reviewers will be printing everything out in color, so do think about that in terms of how busy, how complicated, and what color you are doing these things in. So make sure your legends are also visible if someone prints this in black and white. Proofread and proofread again. Have somebody else proofread it. Why? Because you really do want to be understood. And above that, you want to look competent. You want to look as though you care about the details. So do take that extra time and have somebody else read your proposal for spelling and so on. Punctuation matters, the difference between inviting grammar over for tea and cannibalism. 3 words, 1 comma. Make sure you get it right. Again, attention to detail is super important. I mentioned acronyms earlier. Just beware of acronyms. Don't use too many, because they can be very, very confusing if you're a non-expert, or even just in the reading of a proposal. And one of the things I say a lot is, if a reader ends up confused, it's not their fault as a reader, but yours as a writer. It's your job to get the story across. So make sure you do it simply and clearly, and not in a confusing way. I said it already, but one of the most important things is to make sure that your story gets across. And in order to get your story across here, you need to remember everything that I said here today about getting all of that information in there. But it also needs to look in a way that the reviewers are not finding unpleasant to read and difficult to read. And even more important, you need to register. If you're submitting for April 5th, I hope you are registered at all the places you need to register. So because this is electronic submission, there is a whole lot of stuff you have to do before you submit. And this is a list of all the places that you and your company need to be registered. This process can take up to 6 weeks. Sometimes it can take longer. in a perfect world, it does not take that long, but do be aware this has to be done before you can submit your proposal. And the worst thing that can happen is that you get to submission day and you haven't done this, because that will be, then you can't submit. You literally cannot submit. So make sure that you get this done early. And talking of early, here's my timeline. This is actually NIH's timeline, which suggests that you start your SBIR proposal about 6, 7, 8 months before you submit. Okay, so I'm just leaving this there for those people who want to submit a proposal in 3 weeks. It's not a good strategy, and you'll find it very difficult to do.
Well, for some of you, you may have been doing this stuff over the last 2 years. So if you can encapsulate some of these, you're ahead of the game, but you have to maybe put it down and figure out maybe it's a little closer. But otherwise, perhaps, yeah, that was one of my closing comments, is to never rush a deadline thinking, unless you think never submit something unless you think it's really, 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 really good, because the waiting to the next deadline is usually the best option for you, so you're not waiting another, because of how the deadline and cycles go. It's hard to sometimes meet the next deadline while they're reviewing it and telling you you didn't get it, and there usually may not be enough time to actually submit it with any changes they suggest. So.
Right, and yeah, there are 3 deadlines a year, so yeah, there's always another one coming around the corner.
Yes.
So yeah, absolutely. So, and it it depends. Everybody does things at a different pace. And if you're familiar with NIH proposals, if you know what you're doing, if everything is kind of already tied down, then it can be easier to put this together more quickly. And just 2 more slides here. If you're interested in approaching BBC, if you want to work with us, find out more about what we do, go to our webpage. On the bottom right-hand side, you'll see the link to the SBIR STTR assessment form. If you click on that, fill in the form on the next page, someone will be in touch with you within 2 or 3 days, and we'll set up a phone call and have that opportunity to chat with you. And that's it. Sorry to go a little over time there. That was a lot of information to get across.
That's okay. In fact, the timeline is a perfect one of the closing slides, because we have these webinars recorded. You guys are always welcome, interested in speaking with folks through your process in advance, and have that hard discussion of, are you really ready to submit next deadline? I have a lot of companies I work with that are even working with you guys, and they're working with it for 2 or 3 months, and several times they end up not submitting yet, because they're just something that isn't totally great, and unless it's great, don't submit it, right? So you guys, I think, have discussions with them to say, you may not want to submit because of X, Y, and Z, or just a little bit of X, because it's just not, we don't think it's going to be. I know you guys have those discussions. If a company wants to submit it either way, they're able to. But so that's my comment is especially with NIH and even NSF, there are no topics that specifically you're normally applying to, saying, hey, the one and only time it's ever going to be a topic. These are mostly investigator-initiated opportunities that you're submitting. So you have nothing but a lot of time to think about and put it in the right way. So we hope you take advantage of the webinars, the engagement with the consultants if you can. We are planning to host an NIH/NSF SBIR workshop this coming April in a couple locations. I mentioned that earlier in my slides. So if you decide, if you're still interested in even if you submit in April, you want to come out to those. They're really low cost. The cost is not going to be an issue why you shouldn't go, but maybe if it's the distance, try to go. But let me know. I'm happy to talk to you about whether or not that's something you should go to. We hope you can. And then, so if there's any questions about next steps, please let me know, or you can call the folks at BBC up. I can help initiate an introduction if you don't want to reach out to them directly, and kind of tell you how to get into their system to get that assessment done early. So with that said, I don't know if let me double see if there's any questions. So there really are no questions. I guess we did a great job at explaining everything. If there are any questions after this, let me know. I will be sending an email out, I hope, if not late today, first thing tomorrow, and I'll include the attachments in PDF. And if anybody needs to then look at the online, the recording again, let me know. I'm happy to send that URL to you without having to have you register. So let me know about that, because otherwise we'll post on our website within the next few days, but there will be a registration requirement, and we don't want to have to make you register again. So let me know if you want to view this again. I can send that URL to you. Other than that, we look forward to next week's session is on budgeting. And so if there are issues that you want to have any questions about budgeting, that'll be a great opportunity for you to kind of hear from the source. And again, the BBC folks will be giving that. But again, thank you everybody for today. Sorry I went a little bit over. I actually meant to indicate this session probably will go a little over. And then when I saw how many slides we were presenting, I really apologize for not doing that. But I hope you found today's session enlightening. And there's a great brief overview in a very streamlined amount of time of what it takes to put a good proposal together. But we will go in more depth, of course, at the workshops that we have. So let us know, and thank you, everybody. Have a great day.